Thank you, Andrea. My name is Alessandro Pini. Here you can see my affiliations. Oh, okay, just trying the pointer. My affiliation, I'm professor of biochemistry at the University of Siena, biologist at Ospedale Rescote in Siena, and founder, along with Professor Bracci and Falciani, of this small company, Setlands, and these three institutions are collaborating for the setup of new medical devices for the treatment of sepsis. So, I would like to start my presentation just with a short introduction about sepsis, which you know is a life-threatening, very severe life-threatening condition, which is triggered generally by bacterial infection, bacteria which release toxins and pieces of bacteria which generate a sort of cytokine storm inside the body, together with leaky vessels, clots, and so a sort of inflammatory process out of control, which can generate organ failure and septic shock and death of patients. Okay, here you can see a sort of schematic representation of the evolution of sepsis until the septic shock. Anyway, the most important part of this slide is the numbers. It is calculated that about 27 million of people develop sepsis per year worldwide, and among these 8 million people dies for sepsis. And on the left you can see the commercial value of sepsis. It is not so beautiful to talk about the commercial value when regarding pathologies, but you know how important is the companies which are interested in setting up new treatments, new medicines, etc. for a pathology. And the sepsis market is expected to rise around $1 billion of dollars in the next 10 years. So the interest is very, very high for the treatment of this disease. Also because at the moment, in 2024, no specific therapeutic regimens, or better, medicines exist for the sepsis treatment. Here you can see a schematic representation of a sepsis patient in an intensive care unit, which is initially cured with administration of antibiotics. Of course, the sepsis started with bacterial infection generally, and then many other supportive therapies like fluids, the administration of vasopressors, insulin therapy, and artificial ventilation of lungs, etc., etc. But it doesn't exist a molecular target therapy at the moment. So it is important to have something that can cure directly sepsis when it is diagnosed at the beginning, in an early stage, let's say. Actually, in the last 20 years, let's say, some different medical devices are used in the clinical practice for in extracorporeal circulation system for the purification of sepsis blood from toxins or cytokines. Here you can see the representation of the extracellular circulation system and some some examples of these already commercially available medical devices. The problem with these medical devices already on the market is that no one of these is able to remove lipopolysaccharide from gram-negative bacteria, or lipoteichoic acid from gram-positive ones, or cytokines at the same moment. And this is the main reason for which the use of these devices is not resolutive at all for the sepsis patients. So our project is based the construction of new medical device which can do all these things all together. And the new medical device is based on this antimicrobial peptide called SET-M33, which was isolated in my lab many years ago, actually. And it was initially thought for different applications, for the setup of new antibiotics. Here you can see a selection of many articles that we had in the last years for these different applications, this peptide, which is synthesized in the branched form Professor Bacci talked about before, very stable form, and whose mechanism of action is based on the presence of positive charge on the peptide sequence, which can react with negative charge of biological structure like LPS or LTA, or bacterial membrane just producing bacterial perturbation and destruction of the bacterial structure. So the idea is to use this peptide for conjugating it with a biocompatible resin, and the first prototype that we used was based on agarose beads. So we synthesized the peptide with functional groups. This tile group is used for the conjugate covalent bond with a iodoacetyl group of agarose beads in order to have a stable covalent bond, in order to have a final resin to be used in extracorporeal circulation. Of course, all the experiments that I am going to show you are in vitro experiments. We are not in the clinical evaluation. And different patents have been filed for the application, for the construction and application of this new device. The first prototype was used in vitro. Here you can see some very promising experiments for the removal of LPS from physiological solution or sera spiked with a known amount of LPS. You can see that the removal was higher than 85% with respect to the same cartridge without the peptide. Same thing for the removal of lipoteichoic acids from gram-positive bacteria, and more or less the same thing for living bacteria from the sera of spiked with a known amount of bacteria. So the cartridge worked very, very well, but we had a problem, a very technical and physical problem. The resin is very thin, and we had in the circulation system a sort of counter pressure due to the high packing of the resin. So it was not so suitable for a real application in patients. So we found a new collaborator. This company, Medica SPA, which is based in Italy, near Modena, a company which works with filter for water purification and biological fluids purification, which provided us a new matrix based on this molecule, polyether sulfone polyvinyl pylonidose, so PES for France, let's say, which can be produced in fibers or granules, and a small or large cartridge can be filled with this material, biocompatible materials already used in different applications. The problem was how it is possible to conjugate covalently the peptide to this material without functional groups. The idea was the use of the sterilization phase made by beta race ionizing race, which produce free radicals. So the incubation of the PES with the peptide in the same cartridge, then sterilized with beta race in order to produce free radicals which generate covalent bonds, a specific covalent bonds between the two compounds, in order to have, how you can see here, a stable matrix. Now I skip all the experimentation that we performed for the quantification of the amount of peptide covalent bound, but anyway we had a good quantification of peptide, so we pass to the efficacy of the system. Here you can see an in vitro system with a special machine for circulation using whole human blood or sera or water solution spiked with LPS or bacteria, sterilized with the system of beta rays, and then circulate through the system with a known amount of LPS. And here you can see the kinetic of LPS removal. After 15-30 minutes, the removal of LPS is practically around 100% with respect to the cartridge not conjugated with peptide. So it is practically after the first minutes of circulation, the removal is very, very high. More or less the same thing for bacteria. In this case, it was used Bacillus diminuta, which is the reference bacterium used for this application. And even in this case, after the first 15 minutes, the specific peptide and the cartridge remove all the bacteria. Even if in this case, a non-specific entrapment of bacteria are present in the non-specific cartridge. This is the result of the last months, let's say. A very, very important aspect of this cartridge, which is able to remove also inflammatory mediators and cytokines. In this case, we can see the example of C-reactive protein, which is a very important biochemical parameter for inflammatory process, as you know, but it is also very, very important in its activity for the evolution of inflammatory process, above all when it is out of control. And you can see how the cartridge is able to remove C-reactive protein around 100% with respect to the control cartridge. So the last slides just to evaluate how the sera of sepsis patient after the circulation in this cartridge and the removal of sepsis trigger or inflammatory mediators change. Here highlighted, you can see the amount of C-reactive protein in the serum of patients with sepsis, and after the circulation, you can see the very strong reduction of PCR. So this is not a reduction, this is an abatement practically. More or less the same thing for procalcitonin, which is another parameter used for the evaluation of inflammatory process in sepsis, which is a very important reduction. It doesn't work at all. Okay, even if in this case we have a specific reduction also in the control device. Next step, we are in touch with international companies for the large-scale peptide synth production in GMP condition in order to arrive to the animal models of extracorporeal filtration, blood filtration. Of course, it is impossible to do this with small animals because the extracorporeal circulation machine are not do not exist for mice or rats. So we have a project of experimentation with pigs and then to the clinical evaluation. Here you can see the people working on this project from the Setlands, the Hospital of Siena, Medica SPA, which provided the resin, and the University of Siena, where we work, and Giovanni Cappello here present in the audience, which is the most important researcher involved in this project. Thank you very much for your attention.